In this tutorial, we're going to look at the stages of DNA replication. Let's have a quick recap of the structure of DNA first of all. We know that DNA is made up of nucleotides. One nucleotide is circled here. The nucleotide is made up of a sugar, a phosphate group, and a base. Let's just have a quick look at a more detailed diagram of a nucleotide. So here's the sugar. It's a 5-carbon sugar, which means it's got 5 carbons in it. And because this is a DNA nucleotide, this 5-carbon sugar is called deoxyribose. Now, we haven't shown all of the carbons in this diagram, but we can infer the positions from the shape of the sugar. So at this corner here, this would be carbon number 1, this is carbon number 2, carbon number 3, carbon number 4, and this is the 5th carbon. And every nucleotide has a phosphate group attached to the 5th carbon of the sugar, and the base is attached to the 1st carbon. Okay. So the nucleotides are joined together to make a polynucleotide. Poly meaning many. So many nucleotides joined together in a long chain. And the phosphate of one nucleotide, let's look at this nucleotide here, this phosphate is attached to the 3rd carbon of this neighbouring sugar. And the bond that forms between the phosphate and the 3rd carbon of this neighbouring sugar is a phosphodiester bond. And we know also that the DNA molecule is double-stranded. So we have this polynucleotide strand here, and we have another polynucleotide strand here. And you'll notice that the strands run in opposite directions. So all the phosphate groups are pointing upwards on this strand, and all the phosphate groups are pointing downwards on this strand. We say that the strands are anti-parallel, so they are parallel to one another, but they run in opposite directions. So each strand has 2 ends. The 5' end, written like this, is the one with the phosphate attached to the 5th carbon. Okay, and if we go all the way down to the other end of this particular strand, we can see that this particular nucleotide right at the end doesn't have anything attached to it. If it did have a nucleotide attached to it, the phosphate would attach to the 3rd carbon here, but it doesn't. We say this is the 3' end because the 3rd carbon of the end nucleotide is free. 5' because the phosphate is attached to the 5th carbon at the top. 3' because there's no phosphate attached to that 3rd carbon. And it runs in the opposite direction for the other one. So all the phosphate groups are pointing downwards in this strand. They are attached to the 5th carbon. The 5' end is this end. And all the way at the other end of this strand is a nucleotide that doesn't have anything attached to its 3rd carbon. This is the 3' end. The other thing we know about DNA structure is that the 2 strands are held together by hydrogen bonds between complementary bases on the opposite strand. So A pairs up with T, C with G, G with C, T with A, and so on. This is the complementary base pairing rule. So on the subsequent pages for this tutorial, I'll draw a simplified diagram of this DNA molecule. It will look a little bit like a ladder structure. The vertical lines represent the sugar phosphate backbone of each strand. Sugar phosphate backbone of that strand there. And at right angles, sticking out at right angles from the vertical lines are little horizontal lines which represent the complementary bases. Little gaps in between to represent the hydrogen bonds that hold those complementary bases in place. So before we look at the stages of DNA replication, let's think about why it must happen. So here's my simplified diagram of a double-stranded molecule of DNA. This strand is running in 5' to 3', so all the phosphates would be pointing up on this strand, the left-hand strand, and this strand is running from 5' at the bottom to 3', so all the phosphates would be pointing downwards on this strand. Okay, so we know that DNA must replicate itself, it must copy itself to make exact identical copies of that DNA prior to cell division, so that each daughter cell during mitosis, for instance, receives an identical copy of DNA and an identical amount of DNA. Let's just draw a little picture to remind ourselves. Here's a parent cell, and when it divides by mitosis, 2 daughter cells will be formed. Let's say that this cell just has 1 chromosome. Okay, there's its 1 chromosome. If these daughter cells are going to receive the same amount of DNA, the identical amount of DNA, this chromosome must be copied. And when it's copied, it remains attached until the cell divides, and then each new cell receives an identical amount and identical copies of DNA. So that's why the DNA must copy itself. Replication is the copying process. These DNA strands must separate before the replication process can take place. Let's have a look on the next page at what it would look like when it's separated. So here we've got one section of this DNA molecule has separated, exposing the bases on these separated strands. And there's an enzyme that does this, and you need to remember the name of this enzyme. The enzyme is called DNA helicase. What it does is it unwinds the helix and it breaks those hydrogen bonds between the complementary bases, and it exposing these bases on the strands, the separate strands of DNA. And now these separate strands of DNA will act as templates for the synthesis of new complementary strands. In every cell, there are individual nucleotides. Let's just draw what they might look like. So they might look like little sideways T's. This bit here represents the sugar and the phosphate, and this bit here represents the base. And these are manufactured in the cell, and they're available for when the DNA wants to replicate itself. So it's these that complementary base pair with the exposed bases on the template strand. So for instance, one of these might complementary base pair with this base here. If this base was A, then the base on the nucleotide that's just attached will be T. And weak hydrogen bonds will form between those bases. Let's just have a look at some other examples. If this was C, then the nucleotide that would complementary base pair would have a G base. And if this was G on the template strand, then the nucleotide that joins would have a C base. And if this was T, then the nucleotide that complementary base pairs would be A, and so on and so on. So there's another enzyme that you need to know the name of. And the enzyme is DNA polymerase. And the DNA polymerase attaches to the DNA at a certain point, and it allows these nucleotides to add on, and then it runs up towards the ends of the template strand, adding on the nucleotides and joining them together as well. So as it goes, the nucleotides complementary base pair, and then the polymerase makes the strong covalent phosphodiester bond between those nucleotides, between the phosphate and the sugar of those nucleotides. And off it goes up again and again, and then here again. And it does that going up in a particular direction. So it only runs, the DNA polymerase is only able to run in one particular direction. Remember, we said that this strand is going from 5' to 3'. The new strand will be going in the opposite direction because the strands are always anti-parallel. So this will be the 5' end and this will be the 3' end. And the DNA polymerase can only run in that particular direction. So this DNA polymerase on this template strand will be copying in this direction, from 5' on the new strand to 3'. The other DNA polymerase on the other strand will be going in the opposite direction, running down, adding on the nucleotides and joining them together as it goes. So let's just draw in some of those. So we might have some nucleotides there being added on, and as the DNA polymerase passes over them, they get joined together. The phosphodiester bonds are formed and a new sugar phosphate backbone is formed between the nucleotides. So this DNA polymerase is running in this direction because, remember, the DNA polymerase can only run in a 5' to 3' end direction. This strand is going from 5' to 3'. So this new copied strand will be going from 5' to 3', and that's the correct direction for the DNA polymerase to be travelling. This strand gets copied in fragments and then joined together at a later date. So let's recap. What have we said so far? First of all, we've said that DNA helicase has to separate the strands by breaking the hydrogen bonds. And then each strand of DNA acts as a template for the synthesis of new complementary strands. And then the free nucleotides, free just meaning the individual nucleotides that have been synthesized in the cell, can be used to complementary base pair with the nucleotides on the template strand, making the new strands. And then finally, we talked about the DNA polymerase adding the nucleotides together and joining them with the phosphodiester bonds. So once this section of the chromosome has been copied, then the same series of stages will happen at this section and at this section until the whole chromosome has been copied. And you'll end up with something that looks like this. So we have two identical DNA molecules. And you'll notice that each strand is composed of, each molecule, so we've got two molecules, each molecule is composed of one strand from the old original molecule, as in pencil, and in pink, this is the new strand that was built up during the replication process. So we say, because we've got two identical molecules, each one with a new strand in pink and an old strand in grey, we say that DNA replication is semi-conservative. And that's because the original DNA molecule in grey has been partly conserved within the new molecule. In the cell, the two identical DNA copies, these two identical DNA copies, the chromosomes, remain attached to each other until they are separated during cell division. So they'll look like that. Does that look familiar from your lessons on mitosis? So this is one copy, this is the identical copy, that's what these are. And then they'll remain attached as sister chromatids until cell division takes place, and one will go into one new cell and the other one will go into the other new cell.